Recent advances in genomics have led to a myriad of new targets, and with them the opportunity to drug them using nucleotide-based therapeutics such as oligonucleotides.
The drug discovery process repeats a common workflow to optimize identified hits towards clinical candidates through the design, make, test, and analyze cycle, otherwise referred to as the DMTA cycle. This process is well established for the research and development of small molecules and is now being applied to oligonucleotides.
The first task to design an oligo is to identify the most effective place to target the mRNA. Our starting point is the genetic sequence, and by using computational pipelines, we apply algorithms and machine learning models to predict the most effective and safe oligos.
The oligonucleotide synthesis process begins with a solid phase synthesis, which is well understood and predictable. Our team of chemists program the synthesis of specific sequences, which also enables prediction of the origin of certain impurities. The synthesized oligo is then purified and tested across different assays. Based on the data from this testing, we propose different chemical modifications to optimize the physicochemical properties and avoid toxicities. The oligos then go for profiling using cellular and biochemical assays to test for their effect on the target, from both an efficacy and safety point of view.
Analytical chemistry is an integrated part of the synthesis of oligonucleotides. To monitor the progress of chemical reactions, we provide a walk-up platform of instruments to the chemists to use to check the outcome of both oligo and targeting chemistries. The walk-up platform is based on high-end analytical instrumentation and a high degree of automation. For final batch analysis, we cover analytics from high-throughput characterization of oligonucleotide libraries for in vitro screening to in-depth characterization of in vivo batches, where we apply our expertise to determine the purity of the sample and to verify the composition of the oligonucleotide. To optimize our workflows, we work in close collaboration with the IT platform to build efficient workflows with a high degree of digitalization and automation.
We use next-generation sequencing and transcriptomics to optimize the efficacy, safety, distribution, and mechanism of our oligonucleotide drugs. Furthermore, we are also applying our technology to the development of new pipelines for different oligonucleotide modalities and to validate whether the core project hypotheses are valid across the preclinical models that we use and the human patient material.
The Separation Science Laboratory takes care of the purification of oligo compounds in order to reach the purity required for their intended tests. We handle samples from a few milligrams of complicated molecules for their first in vitro tests to gram scale of high purity oligos headed for in vivo tox studies. As the first group to see the molecules aside from the chemists who made it, we can feed back info about the impurities to help improve the synthesis for future batches. We also help other analytical chemists to prepare for new and different types of molecules as they move into later phases.
Compound management receives and stores molecules coming from chemistry and distributes them into a format which is ready to use for experimental work. By employing brand new dedicated laboratory equipment, we are now able to handle aqueous solutions of oligonucleotides in an assay-ready format through a fast and highly automated process, which makes it possible for us to drive science forward together.
We need to also understand what the body does to the oligo. We are building the infrastructure for in vivo studies, as well as storage and analysis of the PK and PD data generated. In addition, we work on developing the right mathematical modeling tools to accurately predict the human dose and PK/PD of new oligos that we discover.
Oligos also need to escape the endolysosomal pathway to reach and interact with their RNA targets in the cytoplasm or nucleus, so-called endosomal escape. To detect the movement of oligos into the cell, we use microscopy to visualize oligos detected by different fluorescent labeling methods.
While introducing of chemical modification provides great oligonucleotide specificity, stability and safety, one of the main challenges is to deliver oligonucleotides efficiently to the targeted tissue.
ASOs predominantly distribute to the liver and in the kidney. Much lower concentrations are observed in other organs. We are continuing to explore new ways of directing therapeutic oligos to the heart, for example, while reducing the relative concentration in other organs, and thereby improving delivery, efficacy and reducing side effects.
If we can achieve effective targeted delivery of oligonucleotides to specific cells and tissue, we can reduce the dose and the cost of goods as less of the therapeutic is needed. Thereby, we will expand the druggable space, be able to treat diseases in a better way, and ultimately make an impact on patients' lives.
As oligos start to realise their commercial potential, there's a push towards larger volume indications. This new direction provides strong economic and sustainability drivers forcing change from traditional scaled-up discovery processes. In parallel, we're driving pre-competitive collaboration with peer pharma to improve the sustainability of oligo processing and to share regulatory approaches for this new modality.
We continue to make exceptional progress. We are harnessing data and technology to fast forward the pace of our science. We are working with some of the best academics in the field and partnering with companies who share our commitment. We're seeing our portfolio of oligonucleotides grow rapidly.